Okay, good morning, everyone. I'm Shana Kelly. I'm the director of PRIME, the Next Generation Precision Medicine Initiative at the University of Toronto. And it's really my great pleasure to welcome everybody to our symposium today that's being jointly held with the Hebrew University of Jerusalem. This is part of the CANOT Global Challenge program at the University of Toronto. I'll tell you a little bit more about that program and what it is going to enable us to do. But I just wanted to start with a few words about today's event. And let's just start off with accessibility. So to make sure that we can make this event as accessible as possible to people, it will be recorded. The live captioning is enabled, and you can actually display the subtitles if you click on the live transcript function. The chat is actually disabled because we're expecting quite a few people online today, but you can use the Q&A function to leave questions for the panelists. And if you have any problems, you get booted off the Zoom and need to get back on, please email Angela from our team. And I'll just leave this up for a second if you want to jot her email address down. Okay, so we should also just do a brief land acknowledgement. So we would like to acknowledge the land on which the University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and most recently, the Mississaugas of the Credit River. And today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we're very grateful to have the opportunity to work on this land. Okay, so today we are going to have the second session of this symposium. Last week, we had a really outstanding session focused on protein design, protein-drug interactions, and also protein engineering. I thought it was a really fantastic collection of talks. And so today we are going to turn to a different topic, equally as important and front and center in precision medicine, and that's the topic of molecular diagnostics. So we have a really great lineup. The session is going to be moderated by my partner in crime here, Eylon, Yavin. And so we'll have a really nice collection of talks for you, all focused in the area of new diagnostics and new diagnostic technology. So that will be session 2, and then session 3 will be next week, and we'll focus then on drug delivery. So as you can see, we're covering a really nice set of topics here, and it was just wonderful to see how this came together with the two institutions. So before we launch into our scientific program today, I just wanted to say a few things about PRIME and our precision medicine initiative here at the University of Toronto. If you were with us last week, it's going to be the same material that you saw last week, so feel free to tune out for a few minutes. But I'll just briefly talk about who we are, our goals, the things that we're working on, and then I mainly want to emphasize what we're doing with the CANOT Global Challenge program. And so first, University of Toronto, if you're not familiar with us as an institution, we are a large research intensive university. We've got 90,000 students, 14,000 faculty, about $1 billion a year in research spending in the biomedical sciences. So we're a very large research hub, and made, excuse me, even larger by our partner hospitals, which include a pediatric hospital, Sick Kids Hospital for Children, UHN, which is actually a collection of hospitals, Mount Sinai, and then also Sunnybrook. So we have a very large corridor in the medical sciences. So at PRIME, this was founded as an attempt to really collect up all of our talent and research strength from across campus to better align everybody working on areas adjacent or at the center of precision medicine. So drug discovery, disease biology, the development of new diagnostics, these are all core areas of interest for our members. And so this is a cross-campus initiative. We've gathered up about 75 faculty members. Each faculty member brings in 2 of their trainees into Prime, so we have 150 trainees who are part of the effort. And so this really galvanizes our community and brings them together so that, again, we can really leverage our shared research strengths. And so in terms of our goals, really one of the things that we work towards is elevating and better resourcing research in this area. And so we work on behalf of our faculty members to find them additional resources for their research programs to really accelerate the next set of breakthroughs in precision medicine. We really try to empower collaborations both on campus and beyond with institutions across Canada, across North America, and in this effort across the world. We also have a training mandate because we have so many trainees that are part of Prime. We're very focused on training the next generation of multidisciplinary and translational leaders. We also try to create a greater sense of community, again, both in Toronto, Canada, across the world. And then we're also very passionate about building a biotech ecosystem, a stronger ecosystem in Canada and beyond. In terms of the research areas where we do have really critical mass and quite a bit of strength, they're listed off here: protein engineering, high throughput omics, precision diagnostics, drug delivery and nanomedicine, just to name a few. So all very important cutting edge areas, and many of them the subject of this symposium. I just want to say a few words about our fellows program. So this is where we make most of our investments as an initiative. Every year we hold a competition, we have a call for applications, and what we eventually fund are fellows, so either graduate students or postdoctoral fellows, who then get a new collaborative project going on behalf of 2 faculty members who are part of Prime or more. One of our projects here has 3. And so these are all multidisciplinary collaborations. They're high risk, high reward type projects. And then Prime as an organization then tries to build a full project around the fellows and helps the fellows and the faculty teams write grant proposals, get them out the door to build fully resourced projects around the efforts that our fellows get off the ground. So now over to the CANOT Global Challenge Program. So the CANOT Fund has a very historic beginning at the University of Toronto. This was in part funding that came from the sale of the CANOT Medical Research Laboratories. The CANOT labs were where Banting and Best actually made their discovery of insulin. And so this was kind of an original biologics research lab, and then was eventually also outfitting to do some manufacturing of insulin, other types of vaccines and antitoxins. And so the funding that we receive for this program goes all the way back to that initial discovery. And so we wrote a proposal for the CANOT Global Challenges program. We did this at a time where we envisioned people getting on planes and going to partner institutions and getting to know those partners in person. Obviously, then the world changed on us a bit, and so we're doing things a bit differently just during this initial phase of things. And so we prioritized 4 institutions to be partners in this program. Hebrew U obviously is one of them, also UCSF, the ETH, and the National University of Singapore. And so what we'll be able to do through this program is to be able to create opportunities for exchange. So postdoctoral fellows and graduate students will be able to travel to partner institutions to really foster collaboration, learn new techniques. And so we hope to have a really nice exchange of personnel going back and forth. We'll also have a speaker series. We're going to have a program also for undergraduate students. And this is something that we can build on. The University of Toronto and Hebrew U have a strong relationship that's already in place. We have some other funding mechanisms that we'll be able to access. And then eventually we may be able to do things like build joint doctoral programs. I think there's a lot of really interesting things that we can pursue once we get our work off the ground. So since we can't really send anybody anywhere at the moment, we decided to kick off this effort with a series of joint symposia, just so that people in the two organizations can get to know each other a little bit better. And now that we're kind of halfway through this effort, I think this was actually a really great way to get things started, get more people linked up and knowing about the research at the partner institutions. And so, we'll do this for now. And then once the world opens up a bit more, we'll kind of go back over into the mode that we had originally envisioned. So that's the CANOT program. So with that introduction to the University of Toronto and Prime and our CANOT program, I think we are ready to jump into our scientific program. And so Eylon is going to be our moderator today. So he's also our first speaker. So I'm going to introduce him, and then I'm going to hand off the program to him. He'll do all of the other introductions and moderate the discussion. So just give me one moment to pull up my introduction for Eylon.
Should I upload my slides meanwhile?
Yeah, why don't we switch while I'm
okay,
doing
sounds
that.
good. So I just because I see that you have a slide of me there, so should I just ignore it?
Why don't you go ahead and
yeah, okay. okay. Okay.
Okay, and for some reason, okay, when I stopped sharing my slides, my camera also stopped working. Don't ask me why. I'm still here.
At least we can hear you.
Yes, okay, as long as you can hear me, then we're good. Okay, so your slides are there, they look great. So let me just quickly introduce you. Eylon is a professor at HUJI. Let me just give you a little bit of his background. He did his PhD at the Weizmann Institute of Science. He did his postdoctoral work at Caltech in the laboratory of Professor Jackie Barton. He joined HUJI through the School of Pharmacy in 2006 and has ever since run a really exciting research program in the field of nucleic acids chemistry and also the applications of nucleic acid chemistry. And today we'll hear about his work in diagnostics. Published lots of great journal papers and was recently elected as the president of the medicinal chemistry section of the Israel Chemical Society. And quick fun fact about Eylon, I think we'll hear more fun facts today, but we are academic siblings. So we both worked in the same lab at Caltech, and so that was our initial point of connectivity. And I can't wait to hear what you've been up to, so please take it away.
Okay, thank you, Shannon. Thank you very much. Okay, great. So by the way, by moderator, I just want to mention that each one has 20 minutes. So I'll also try to keep the 20-minute time frame. And there will be no questions after the talks. We'll just have the open session at the end. So I just wanted to note that. So great. So I'm going to tell you today about how we use a term that we call FIT-PNAs to detect RNA in human disease and in malaria. And to start with, I'm just going to explain two terms, general terms. One is PNA, which is peptide nucleic acids. These are DNA mimics. As you can see, they have a peptide-like backbone, where on this peptide backbone, you have the nucleobases that mimic the DNA structure. And what we like about this molecule is that when we make it in the lab, we generate it, and the PNA is very stable, so it doesn't degrade. And because it's a neutral molecule, it also binds readily to DNA and RNA. And generally, it's not toxic. has low toxicity. So these are kind of nice starting points to make these DNA mimics relevant to biological applications. But in the terms of sensing, what we've done in the last years is to use a concept that was developed in the early 2000s by Oliver Seitz from Germany, which is called forced intercalation, or FIT-PNA. And the idea, I think, is pretty cool. It's pretty simple, but pretty cool. And the idea is that you replace one of the nucleobases in the PNA with a cyanine dye. An example for that you can see down here, which is the thiazole orange. And the idea is that when the FIT-PNAs in single-strand form, it's pretty much non-fluorescent. That's due to the free rotation of this monomethine bond. But once the FIT-PNA binds an RNA, complementary RNA sequence, it forms a duplex, obviously. And then this dye has nowhere to rotate, and it's emissive. It becomes fluorescent. So it's in fact like a turn-on fluorescent probe for RNA. But what's even cooler than that is that the fact that you can actually design it to sense mismatches or point mutations. So imagine that at a position that is adjacent to the cyanine dye, you have a SNP, then the region around this duplex is not complete. There's a mismatch, and then the thiazole orange is much less emissive. So we were thinking this seems like a cool idea to detect RNA, but we were also thinking about to use it in biology. And one of the maybe disadvantages of this thiazole orange is its photophysical properties. So the thiazole orange excitation is a bit over 500 nanometers. And there are a lot of biomolecules that absorb and fluoresce in this range, like flavins and cofactors. So that pretty much, can disrupt this fluorescence. You can see here also a spectrum of absorption of hemoglobin and oxidized hemoglobin. So in fact, if we thought that if we would be in the red region, or even better in the near IR region, we would have a much more suitable probe for biological samples. So a few years ago, we developed a new probe. We call it BISQ. You can see the structure here, and you can see how similar it is to thiazole orange. It's simply made of two quinoline that are connected through this monomethine dye. And this simple change in structure actually results in more than 80 nanometer redshift. So we were very happy with that. And all the data that I'm going to show you today is actually using this BISQ monomer. Okay, so what can we detect? What kind of RNA? So in humans, obviously, a very good thing to do is to detect cancer. And there are many genetic alterations in cancer. Specifically, we looked at two. Of course, we can think of others, but for this talk, I'm going to talk about two alterations. One is simply gene amplification. So many oncogenes are overexpressed in cancer. So we've looked at a couple of them, or even more than a couple, on long non-coding RNAs that are mentioned here. We've also looked at single point mutations, and I'll show you an example of the KRAS gene. So as a proof of concept, we really started with the KRAS gene. And this is an oncogene that when mutated, it has like a prolonged activation, causing cell proliferation. And interestingly, you find the KRAS mutants, especially in the 12th codon, very much abundant in many cancers. Just to give one example, is in pancreatic cancer, over 90% of patients have point mutations in the KRAS gene. So again, as a proof of concept, what we did was we designed a few FIT-PNAs with our BISQ dye. And this shows you an example of actually the successful amplification of this technology to detect a SNP. So here you see the FIT-PNA probe with the BISQ, and just adjacent to the dye, we have either the wild type sequence or the mutant sequence. And in fact, what you see is that when you simply incubate the PNA probe, and this is something that I didn't mention before, but the PNA has a very simple peptide on it, just 4 lysines, and that seems to be the job, to do the job in terms of cellular uptake. So we just incubate the PNA probe with cancer cells in culture. And these cells, the PANC1 cells, have the point mutation that we're aiming at, the G12D KRAS SNP, and you see that they light up, whereas other cancer cells that are wild type in KRAS are pretty much silent. So we were very happy with that, and that was like the first proof that these kind of probes can work. I'm just showing one more thing here in this table. You don't have to look at all the data, but I'm pointing out to something which is the quantum yield of these FIT-PNAs, and they're a bit over 20% quantum yield, which is pretty high. There's quite bright probes. And I'll come back to this because I'll talk about some new generation FIT-PNAs, and I'll mention the quantum yield. Okay, so moving along, we next asked, can we really detect overexpression of oncogenic RNA? And in this case, we collaborated with a surgeon, which is sitting in Tel HaShomer, it's a suburb near Tel Aviv in Ramat Gan. And Professor Avi Nissan is a specialist in colorectal cancer, and he does many surgeries related to the abdomen and that area. And what you see here is just a profile of CRC patients where they took biopsies from the cancer tissue, and you see nicely how this CCAT1 colon cancer associated transcript, which is a long-coding RNA, is highly expressed in these patients. So we thought it would be a nice biomarker to detect in cancer. So we designed our FIT-PNA probe for this CCAT1 FIT-PNA, and you can see the nice growth in fluorescence as you make a duplex. The fluorescence increases nicely. And then again, we tested this in cells, okay, simply by adding the FIT-PNA to the cells. So these are cells that highly express CCAT1. These are melanoma cancer cells that do not express CCAT1, and they're pretty much transparent. Okay, so we could show that we can detect SNPs, and we can show that we can detect overexpressed RNA. But really one of our goals and our fantasies was to do something related to fluorescent guided surgery. So just to mention very, very briefly, today there are in some clinics a methodology where you inject into a patient that is supposed to go cytoreductive surgery. You inject a fluorescent near IR marker, IV, and then if it's a targeted fluorescent probe, then it accumulates in the tumor. And then the surgeon wears these really cool goggles where he can see not only the tissue, but he can see where the dye fluoresces in the cancer tissue, and then he can more precisely remove like very tiny points of tumors, as you see in this image. With this idea, we said maybe we can just do something very simple as a proof of concept, is to take fresh tissue, cancer tissue from surgery, just put the tissue on a slide, spray our FIT-PNA on the slide, and image it by microscopy. So we're working with Avi Nissan, and he's giving us samples from tumors and from adjacent normal tissue. And you can see here the tissue in PBS, and we simply take this tissue, put it on a slide, and spray our FIT-PNA. And we were very, very happy to see that it actually works. So what you see here is an example of a FIT-PNA targeting the CCAT1 RNA. And specifically, we're talking about here a metastatic tumor. So this is a peritoneal metastatic tumor, which is generated after somebody is sick with colorectal cancer. The tumor simply metastasizes to that region. So this is grade 4 CRC. And you can see that by simply spraying our probe on the tissue, in a matter of minutes, we see a very bright fluorescence. We also see it for this mRNA, but it's not so distinct as for the CCAT1 RNA. So this is a very simple procedure, right? I'm just taking my probe, putting it in buffer, and spraying it directly on the tissue. And it's the fresh tissue. I'm doing nothing with the tissue, no fixation, no freezing, no nothing. Okay, and just to show that this works, we did a few very important controls, which is to spray the probe on normal tissue. So this is bariatric surgery, just normal colon tissue. We see no fluorescence. And we take a non-targeted FIT-PNA on the tumor, and it doesn't fluoresce. And the last thing I want to mention about this is that when we take the tissue, we cut it into two, and half of it goes for imaging and half of it goes for PCR. And you can see that in all the tumors that we take from the surgery, they all have high expression of CCAT1 and KRT, whereas the bariatric normal tissue has no negligible expression of these biomarkers. Okay, so the last thing I want to mention about this is that we can actually also, we've done some 3D imaging into the tissue. So obviously we spray the PNA topically on the tissue, but we see that it penetrates over 200 microns deep into the tissue. So both for both biomarkers, we see tissue penetration. Okay, this is kind of recent data that we have for another long non-coding RNA, and now we're looking at breast cancer tissue. And again, we see some nice fluorescence by spraying the FIT-PNA for hot air 1, which is also oncogenic long non-coding RNA. And the scrambled sequence shows no fluorescence. But this, we still need to repeat these experiments and see that they're more robust and reproducible. But I'm just showing it to show you that we're not limited to one type of cancer. We can think of other types of cancers. Okay, moving along. What's more recent now in the lab is that we have now created very bright FIT-PNA probes. And we're very excited about this because what we've done is we've introduced 2 cyclopentane rings on the PNA backbone. This is in collaboration with Dan Appella from the NIH. And the idea behind this is that these cyclopentanes give a sterical hindrance to the backbone. And then we hoped at least, and I'll show later, that this sterical hindrance will increase the rigidity of the duplex, making the bisque even more emissive. So here you see the data, and I think it's very promising because it shows that when you have a standard, so this is on a model FIT-PNA, we call it now CPP FIT-PNA, cyclopentyl FIT-PNAs. So we took a very simple model of 12 mer PNA, and what you can see is that when you have no PNA, so this is the standard FIT-PNA, you see the fluorescence down here in green. When you add one cyclopentyl, the fluorescence goes up. And when we add 2 cyclopentyls, the fluorescence even goes higher. So this is very nice. We can see it in both sets. And as I mentioned before, in terms of quantum yield, going from a standard FIT-PNA to one that is substituted with 2 cyclopentyls, we actually double the amount of the quantum yield. So here going from 18 to 33%, and here from 20% to 39%. Okay, so what was even cooler, and we were very happy about this, that in fact, in some cases, we get a very nice mismatch discrimination. I just want to give one example. So it's not for every mismatch that is around there, but for just one example to show you, to emphasize this, in a set of one of these CP FIT-PNAs, you can see that going from the fully matched sequence here to the mismatch, which is a TT mismatch, the standard FIT-PNA gives only a threefold discrimination between a full match and mismatch. But whereas the cyclopentyl modified FIT-PNA gives a 15-fold. So we're very excited about this because now we can think of really certain mismatches. For example, the BRAF oncogene that has a very known V600E point mutation has exactly this TT mismatch. And we hope that we can show that our FIT-PNAs with cyclopentyl are highly sensitive to this SNP. Okay, so we've now started to make real PNAs, FIT-PNAs, in this case to detect ovarian cancers. And what we've done, we've introduced a short peptide. It's called CLIP6. And you see here the sequence of CLIP6. And we've used this peptide in the past in order to show that we can actually have very effective uptake of PNA into cells. This is glioblastoma cells. And we figured out that this uptake is a non-endosomic uptake. So it's like maybe even a diffusive kind of an uptake, which is great because if it works that way, then the bioavailability of the FIT-PNA would be even higher. So we made a whole series of CP FIT-PNAs with CLIP6, and we're now testing this in cancer cells and so on. And I just want to show shortly that basically we see that indeed these now CLIP CP FIT-PNAs detect ovarian cancer cells. They light up, and even we're even more happy about that. It seems that the CP introduction of the cyclopentyl makes a big difference. So here you see ovarian cancer cells that are light up with the cyclopentyl, and those that without the cyclopentyl are pretty much non-fluorescent. Okay, how much time do I have, Shana?
You've got another couple minutes.
Couple of minutes, okay. So I'm just going to go quickly into this. I'm going to kind of skip all the introduction. What I want to show you very briefly is that we're also trying to detect point mutations in malaria. So there are two types of very known drugs, chloroquine and artemisinin, that the parasite that causes malaria has developed resistance to these drugs, and the resistance is based on point mutations. So now a few years ago, we discovered that if we take a PNA and attach a short cell-penetrating peptide to it, the PNA goes into the red blood cells, into infected red blood cells, and it actually can downregulate gene expression in malaria. So after that work, we decided that we want to look at detection of these point mutations in certain parasites that harbor these point mutations, that they would be drug resistant. And I'm going quickly just to show you an example. So I'll show you this example. Oh, the first one actually. This just shows you that if we can design a PNA that detects a point mutation in the gene that causes a drug resistance to the best drug today against malaria, which is artemisinin. And you can see that by simple incubation of our FIT-PNA with the malaria, with the infected red blood cells, we see a very distinctive difference between the population of those parasites that harbor the SNP and those that don't harbor the SNP. And you can see this also by microscopy. Basically, what we're trying to do now is to develop this technology with the CP FIT-PNAs. We're trying to design a kit. I don't have time to talk about it, but we're working on this. and hopefully we could get to an actual kit where we would take the blood from a patient, add it to the streptavidin coated plate with our biotin labeled FIT-PNAs, and detect it in situ just simply by reading the fluorescence of on a plate reader. So this is still in works. I'll skip this, and I'll just summarize that I've shown to you today that we can use FIT-PNAs to detect SNPs and overexpressed oncogenic RNA, also in cancer cells and also in fresh human cancer tissues. And then I showed you how we can now develop very bright RNA sensors, the FIT-PNAs, and hopefully we can develop these into a diagnostic kit for malaria, associated drug resistance, and explore the new technology for other types of cancers, also in cells and also in tissues. And with that, I'll just thank my current group members, the previous ones that worked on the projects that I mentioned, and my collaborators, Professor Reuven Reich on the ovarian cancer, Professor Yavin with the colorectal cancer, and with malaria, Professor Ron Tchaikovsky. And thank you very much.
Great, thanks, Eylon. So I'm going to now hand off to you, and you can take it away.
Sure.
And I just want to echo what you said, that you were right on time. We should just keep all the speakers on time so that we have plenty of time for the discussion at the end. There's already lots of questions coming in.
Okay, great. I can try to give like a sort of a cue if the speakers want, like 2 minutes before. Anyhow, so I'm going to turn off my presentation. Our next speaker is Professor Keith Pardee. I hope I'm pronouncing that correctly. I'll say a few words about Keith. Keith is a synthetic biologist. He's at the Leslie Dan Faculty of Pharmacy and University of Toronto, and he's working on drug development and disease diagnostics, and actually working on cell-free systems. So I guess we're going to hear quite a lot about these very cool systems that one can develop really interesting tools to figure out things like diagnostic tools and drug manufacturing and all kinds of cool stuff. I'll just mention one fun fact about Keith, that he's sort of a carpenter. I mean, he likes restoring old furniture. So I'm glad that you have time to do that, Keith. That's great. And go on doing that.
Yeah, limited.
And Keith, you can go ahead and put up your presentation. And the title of his talk is Responding to Outbreaks, an Emerging Role for Synthetic Biology and Low-Cost Hardware. So
great. Well, thanks, Eylon. I really enjoyed that. I'm really fascinated by the material you just presented. Very cool.
Thank you very
lots
much.
of ideas thinking now. So
okay, great. Wonderful.
Thanks for the introduction. As Eylon mentioned, our lab works in the area of synthetic biology, and specifically cell-free synthetic biology. And we do this so that we can create in vitro platforms that are abiotic, biosafe, so that we can take genetically encoded tools out of the lab and sort of into the world. And we do this for portable molecular diagnostics, as well as the portable production of protein-based drugs, which usually require a cold chain. So today I'm going to give you a little bit of a background on the platform that underpins the type of work we're doing. I'll then talk about some of our applications in the diagnostic space, focused on our previous work on Zika and their sort of current efforts on SARS-CoV-2. And then I'm going to touch on a new paper that we just had come out that's sort of a new way to use our cell-free systems. So as I mentioned, we're dedicated to sort of building these decentralized tools for healthcare. This is in the form of gene circuit-based diagnostics and also the portable manufacture of protein-based drugs. These are relatively new ways to do these things. And so there's lots of fun things. It's sort of the space is open. It's lots of fun to work in this area. And we do this primarily because these new platforms allow us to do this in a portable way, but also in a fairly low cost way that has the potential to improve access. And so we have projects in the area of food security, emergency preparedness, general health, as well as national security. So I'll start off giving a bit of a background on the system. So we use cell-free systems, which are basically these cell-free enzyme mixtures that contain the enzymes of transcription and translation. And primarily we use E. coli as our sort of source organism, but we've also used HeLa cells, tobacco cells. You could sort of use any system. We then add a genetically encoded tool that carries either the DNA encoding sensor so that we can detect an analyte of interest and then produce an output, or to produce a drug of interest. And then we can freeze dry these components so that they can be distributed without a cold chain. So it's sort of improving distribution. And then in one embodiment, we embed these enzymes into paper discs. These are just 2 millimeters across. They sit at the bottom of a 384 well plate. Both have the enzymes, but only the disc on the right has the DNA encoding for green fluorescent protein. And then you incubate at 30 degrees for 20 to 30 minutes, and you start to see GFP express. And in the other sort of format we use, there's no paper involved here. Here it's just we freeze dry the cell-free system into pellets. And then when you need them, you rehydrate and you get the expression of your protein. In this case, of course, it's just GFP, but we do a lot of antibodies, vaccines, antivenoms in this sort of format. And so the subject of today's talk is on diagnostics. So we'll be mostly talking about the paper-based format. And so what I just shared is basically the operating system for what we do. And then of course, we now need to specify function. And so for that, we use often RNA-based components. And one of the ones we use a lot is a toll hold switch, which was developed by Alex Green, who's an ongoing collaborator. When he was doing his postdoc at the Wyss Institute with Jim Collins and Peng Yin, he developed toll hold switches, which are RNA hairpins that sit at the 5' end of an mRNA. And they sequester the ribosomal binding site and the start codon in a way that the ribosome can't interact with them. And then this region marked AB is essentially the sensor surface, and it can be designed to detect virtually any sequence. And so it's basically complementary to your pathogen of interest, this genome. And how it works is in the presence of the correct sequence of your target here, the A star region binds to the single-stranded toll hold switch that is hanging off here, and then B star binds to B, and you get this basic linearization. And now the ribosome can bind, and you get expression of your gene product or your reporter. And so this gives us a way to essentially rationally design diagnostics. We needed other features to make this useful out in the real world. And so for that, as I mentioned, we turned to freeze drying for distribution without refrigeration. We also wanted for users to be able to see what's going on at the molecular level with their eyes. And so we developed, without hardware, so we developed colorimetric outputs that can be detected by eye. And for applications where you may have a lot of patients, you want to do screening or you want to quantify, we developed low-cost hardware to help complement this. And so in our first paper, which is shockingly a long time ago now, 6 years or so, we developed this color-based output. So the paper discs turn from yellow to purple. We also developed printed arrays, so you, in theory, could distribute multiple tests in a piece of paper that's sort of the size of a postage stamp. And then we developed our sort of first generation electronic reader, which could read 12 reactions at a time. And then we demonstrated these sensors in the detection of genes responsible for the antibiotic resistance of 4 different antibiotics, as well as strain-specific detection of the Ebola virus. And this was exciting at the time because this was the first time that this type of gene circuit-based sensor could be deployed out of the lab in its abiotic format, but they weren't sensitive enough to be clinically relevant. And so we spent the next year sort of solving that challenge. I finished my postdoc and I came to Toronto, and about 3 weeks and this was January 2016. And so about 3 weeks in, Jim Collins, who was my postdoc supervisor, called Alex Green and I, and basically with the Zika outbreak happening, suggested, we thought we'd taken the system far enough to contribute to the Zika outbreak. And so along with a lot of other people from 7 different institutions, we sort of went and tackled this. And so Alex had an algorithm that interrogates the genome of the pathogen, looking for low secondary structure where these primers or probes could interact with. And then we just take the complementary sequence and place it into the toehold switch region here. And so we built 24 sensors for each target gene. And the cost of these sensors is quite low, and it's relatively fast to develop. To solve the sensitivity issue, we placed Melina Fan, who's from Adgene, developed an isothermal amplification loop. And basically what it does is it's a primer-directed process where the primer is specific to the RNA genome of the virus. Through reverse transcription, you get this RNA-DNA duplex. And then in the presence of RNase H, and this is a one-pot reaction, RNA is degraded when it's bound to DNA. So now we're left with a single-stranded piece of DNA. And there's a second primer that binds with that has a T7 RNA polymerase promoter. And so through double-stranded DNA synthesis, we get this piece of double-stranded DNA with a T7 promoter. And so this is basically a copier, right? And it's making millions of copies of our target RNA now, which is what we use to rehydrate our paper discs. And so when we did this, we could now detect into the low femtomolar range, which is clinically relevant. And in the time since this paper came out, we've brought that down into the low attomolar range. And so this platform allowed us to detect all the global strains of the Zika virus at the time, but still distinguish from other endemic mosquito-borne infections like the dengue virus. And then Guillaume Lambert, who's now at Cornell University, developed a very sort of the first use of CRISPR in diagnostics, and it could detect using PAM, the difference in PAM regions, discriminate between the strains of the virus. So that, which of course was important because of microcephaly and other neurological issues. And sort of what was really I guess, like while we were very excited about Zika being able to detect, is that we just basically established a programmable platform that could be pointed at any pathogen of interest. We were then fortunate enough to be able to take this diagnostic to Latin America. And we collaborated with 8 labs in 5 countries and expanded our sort of envelope of diagnostics to include chikungunya as well. And the mandate of this project, which we're just writing up now, about to submit, was to validate the molecular tools with patient samples. And one thing we hadn't counted on was that we would also need to become a hardware lab. I'm a molecular biologist by training, but our partners in Latin America didn't have 384 well plate readers. And so we had to basically build one ourselves. And in the end, it was actually became a strength for the project. And so we developed this low-cost plate reader that can handle 384 well plates. It's incubated, it's automated. It has a user interface that's very similar to any sort of conventional plate reader. It's camera-based, so it's very inexpensive. It costs about $500 to build. And we can easily discriminate between positive reactions and negative reactions. And there's also an algorithm that automatically bins reactions into positive and negative. And this was the effort of 2 very sort of superstar students, Sarai and Livia, who've since graduated and gone on to start a little startup. And I'm extremely sort of proud of their hard work. They're now selling these devices. It's just the first few months of doing that, but it's coming along. And so all the data that we collected in Latin America was done on those devices. And so we were down in Latin America twice, once in Ecuador to train our collaborators on molecular tools and hardware, and another time to start the patient trial in Recife, which was sort of the epicenter of the Zika outbreak in Brazil. And so I'm going to show you guys some of the data that we collected, or I guess the most important data that we collected. So the data on the left was measured using the Plum reader, that low-cost camera-based reader. And the data on the right was collected using conventional RT-qPCR, using the CDC diagnostic for Zika. And so you can see here we wanted to test for specificity. So we had a panel of other endemic arboviruses from Latin America and the Zika virus, the American strain. And you can see the colored paper discs. This is just looking down the holes of the wells. You can see that only the Zika by eye was purple. The others were all yellow. And that is reflected here with the measurements on the Plum reader. And the RT-qPCR, not surprisingly, found the same results with this is a high concentration of virus in all circumstances of cultured virus. We then wanted to determine the sensitivity of the assay, and so we titrated cultured Zika virus from 10 to the 4 down to 10 to the 3 plaque forming units per mil. And again, we could detect, as you can see by eye or through the quantification on the Plum reader, down to 10 to the 1 plaque forming units per mil, which we were really pleased to see match the sensitivity of the RT-qPCR diagnostic, which could read basically the cutoff threshold is 38 for CT values of 38 for the trial for the Zika RT-PCR test. So we then went on and did a patient trial with 270 patients and found an accuracy of about 98%. So we're pretty pleased. And this sort of set us up this was the fall of 2019 when we finally finished the Zika work in Latin America, which was good timing for us in terms of being able to have a platform ready for the COVID outbreak, which has been the focus of many people's research over the last year. And so we have a few projects. The one I'm going to talk about today is the development of toehold switches to the SARS-CoV-2 genome, and the detection, and then basically the creation of surge capacity kits that could be deployed. This grant was written in February 2020, before the Zika, or before the virus, the SARS virus had left China, and so we were still sort of thinking it wouldn't be coming to Toronto, and also being able to make kits that could be deployed for fabricating these tests, and with a focus on low and middle income countries, and basically trying to develop PCR independent diagnostics that didn't require the consumable supply chain or the instrument supply chain for PCR. And so I'm going to focus on just the development of the sensors themselves. This work, this is sort of the original team for the grant, and it's since expanded to include several other countries. The work was led by Masoud and Margot, who are incredible postdocs in my lab and who have been doing, had a very busy year. And so what we did was, with Alex Green again, we developed sensors targeting 6 regions of the SARS-CoV genome in 4 different genes: open reading frame 1B, the RDRP, the envelope protein gene, and the nucleocapsid gene. Okay, 2 minutes, got it. Okay, so using the same workflow, we developed toehold switches again, where computationally and then using cell-free systems and isothermal amplification, we developed these color-based switches. So I'm going to speed ahead. Tight timeline here, and I went anyways, so we were able to successfully develop sensors for each of these regions very quickly. So that was great. And this is just done using LAC-Z outputs. So the brick colored lines are in the presence of synthetic RNA. We got detection down to 10 copies per of RNA per reaction. And then we did a patient trial, I'm speeding up here, where we were able to detect all of, this is with optical outputs, up to samples, patient samples up with up to CT values of 31, except we missed this one, which is 31.9. And we also had one missed negative. So we had this work is ongoing. We're continuing to improve our sensitivity, but we basically have a 90% detection here with, which was blinded. And then one advantage of doing work in remote regions is that the needs at the end for the end user are highlighted. And so we wanted to figure out, well, how could we put gene circuit-based diagnostics into the hands of users in a more reasonable, practical way? And so a paper that just came out in February, we basically merged gene circuit-based diagnostics with what's all sort of very ubiquitously available glucose meter based diagnostics. And so this takes advantage of established manufacturing and distribution channels. And the work was done by Evan Amalfitano, who's did a ton of work. This took a lot of time to get this to work. And basically he did this by replacing the optical reporter gene with a glucose generating enzyme that converts an inert substrate into glucose. And then it can be read on the diagnostic. And so we use the same sensors that we used to create this yellow purple color, but then just switched to glucose. And here you can see, so this data was all collected on an $11 glucose meter, where glucose is measured milligrams per deciliter on the left. We got detection down to the low attomolar range. We could also, we had high specificity against other respiratory viral genomes, and in a very small test run, we were able to detect COVID positive patients and discriminate from healthy patients. And this was a little incubator that we built. And this is just basically showing that our data in the incubator was very similar to the data in a commercial plate reader or a thermocycler. And so I will just wrap up. I'd like to thank my team, which has been doing amazing work over the last year, especially Margot, Masood, and Evan, Alex Green, who's now at Boston University, and his team, and the Zika trial teams, especially these folks here, and Linda Marpena, who helped run things in Brazil and the glucose meter. So with that, I will pass it back to you, Eylon. Thank you.
Okay, great, Keith. Wow, that was amazing, amazing talk. Thank you very much. We're pretty much in time. We're doing pretty well. So we're going to go to the next speaker, which is Professor Galia Blum from the School of Pharmacy, Hebrew University, my good colleague, close colleague. Galia works on, Galia, you can put up your presentation meanwhile. Galia works on designing and developing very cool chemical probes for studying and sensing proteases. So we're going to hear about that. Galia's fun fact is she's pretty much a semi-professional basketball player. She used to be a professional basketball player, like in the national Israeli team, which is quite amazing. And since then, I think it's, what is it, Galia, more than around 30 years. No, I'm not far from that, right? Time flies. So she's been up to it, and she still goes on, and once in a while it gets injured, but she doesn't give up. so anyhow, so Galia, the floor is yours, and looking forward.
Thank you, Eylon, and wow, these are amazing talks. So thank you for inviting me to this prestige set of scientific findings here. Wow. So I want to share with you some of my research that are basically tools to detect diseases using protease activity-based probes. If we're talking about proteases, then I want to tell you just a little bit about the proteases, so we would be on that you will understand what's going on. So proteases are generated in cells as inactive enzymes, and they undergo processes of activation because what they do is cleave other proteins. And if they do that uncontrollably, then they could be very harmful to the cell. So the proteases themselves detect their substrates as a sequence, as their sequence, and different proteases cleave the same sequence. I don't know what's going on here. Different proteases detect the same sequence, and the single protease could detect several sequences, and you have like this consensus sequence that are detected by certain proteases, and then they cleave these substrates. The enzymes, these proteases, could also be inhibited by endogenous inhibitors, and that basically makes it very, very difficult to try and detect when an enzyme on one of these proteases is actually active or when it's inactive. So the set of proteases that I'm interested in are called cathepsins. These are cysteine proteases, meaning they have a cysteine in their active site, and they're highly overexpressed in cancer. But if I want to say specifically in cancer, they're overexpressed in the stroma of the same person. That means in the immune cells of that person. And name the specific ones that really highly express these proteases are macrophages. But when these proteases are expressed usually inside the cells, in the lysosomes of the cells, they could also be secreted out of the cell, and then they could degrade the extracellular matrix and actually enable these tumor cells to be metastatic. So it's very interesting that there's quite a bit trials into inhibiting these enzymes, but what we want to do in our research is mainly use these enzymes to target our contrast agents and enable us to detect this specific lesion. So before I go into these probes, the first set of probes that I generated still when I was doing my postdoc at Matt Bojo's lab at Stanford, we generated these fluorescent probes that bind to proteases, the cysteine cathepsins, and we injected them into the tail vein of mice that were bearing mammary fat pad tumors. And what you can see here very nicely is that the green cells that are the tumorigenic cells expressing GFP, those somewhat express high cathepsin activity, but the ones that are coming from the stroma that we later faxed out to see that those were actually macrophages, those really highly overexpress this red marker, and you could see this by fluorescent microscopy. So I want to share with you a little bit of how we generate these activity-based probes. They're generated as this module. We could actually play around with each in the set of these modules. They have a tag so we could detect this. And we use different types of tags. I'll go into this through the lecture. They have a recognition element, it's usually a short peptide that is recognized by the enzyme, and this drives selectivity. But also we have what we call an electrophile or a warhead, which actually enables these probes to covalently modify the enzyme. So what we would get is basically something like this. We have our probe here, the enzyme, and this is a thiol of the cysteine protease. It will try to cleave this substrate because it recognizes it as a substrate. It will actually target this most electrophilic point, and it will generate this covalent modification, this thioether bond. And now what we're resulting with is an enzyme that's covalently modified by the tag, and then we could detect this. But if you notice, this is a covalent modification, so it's different than what we usually see, like amplification, that we have a substrate that gets cleaved and on and on. This binding of the enzyme to the probe actually also inhibits the activity. Also, it releases this active leaving group that we later then use for our purposes. And these types of probes, actually, the activity-based probes were there when I started, but when I got to Stanford, we decided on generating a new type of probe. And the new type of probe was basically an activity-based probe that has a quencher at the end. And this resulted in a quench fluorescent probe. So these probes don't fluoresce till they actually bind the enzyme. When the enzyme would bind, the quencher would be removed and it would fluoresce. And this type of structure would actually enable real-time imaging, because you would only see this probe when the enzyme binds to it. But when the enzyme's not bound to the probe, then we don't see any fluorescence. So that you can really tag when the enzyme is active and where it's active immediately. Because if we go with the previous version, it was fluorescing all the time, and then you need to have some kind of process to get rid of the fluorescent probe. But here, the free probe is not fluorescing. So this is how it would look. You have a probe here on the right. This is a quench fluorescent probe. This is a crystal structure cathepsinel with a cysteine active site. Then you'd have the probe bind in. It's recognized by the enzyme. The cysteine will actually attack that acyloxymethylketone, that electrophile. It will release that acid, and now it will light up. So we have like this turn on fluorescent. And originally we generated these types of probes into cells. We saw that it works very nicely with cells. But later on, we actually applied this into animals, and we were able to show that actually this detects cancer. When I arrived to the Hebrew U, I decided to actually make this out into a platform. So we could actually do all kinds of things, as I was saying. So I could base on the quench fluorescent. We could also put a cap and put different tags in. So what we've been doing here is using optical tags, so more fluorescent tags, and I'll talk a little bit about that. And CT tags, because the idea was that we could actually use this later on for in humans. So we need something that we could detect deep into the tissue. And then I wanted the detection to actually be using CT, so we could actually go deep. And this is something that is actually used in hospitals all around. And then why only in cancer? Actually, we have different types of enzymes that we could use. And we also have different diseases that are highly inflamed, like atherosclerosis. Also, that's an inflammation of our arteries. So basically, we could actually use these types for more than just what we were talking about. But I thought that the coolest idea was actually to generate something that's called a theranostic probe that could not only be used for detection, but also for therapy. And if we think about that, we tried to take this probe, and instead of just attaching a regular fluorophore in there, we could attach what we call a photosensitizer. So these are light fluorophores, but when you have light shown at them, they generate reactive oxygen species that actually kill the cells. So this was the first design which we started on with, having that same design with the quench probe. The photosensitizer now would be inactive because it would be quenched. But as soon as the enzyme comes in, the quencher would be released. Now it would be light sensitive. That would generate reactive oxygen species and actually kill the cells. And we would get basically 2 in 1. We'd get a probe that could be used for therapy and for diagnostics. So that's a theranostic probe. And I won't go through all the development because this is quite old data, but we designed these compounds, we modified the wavelength that these probes were working on. So we had near IR probes. They work over 700 nanometers. And we saw that they actually labeled the enzymes, if they're cell permeable, that they're quenched, and so on. We do all that before we went into the animal experiment. And this animal experiment, what we did is we actually had two tumors on the back of the mouse. We inject these compounds into the tail vein. As I say, they're quenched, so you don't see them in the body after 10 or even 1 hour, 10 minutes or even 1 hour. But after a few hours, they start accumulating in the tumors. You could see them fluorescing. And as I said, these are covalently modified, so they attach there. Then we shine light only at one of these tumors, what's the one that is seen here on the left. And then we kept on following these mice, and we gave another light dose after 16 hours. And as control, we basically have the same photosensitizer, but was lacking all this targeting weight and the covalent modification. So we saw that worked very well. And then what we saw later on is that when we treat these tumors, we actually see that only the mice that were treated both with this compound and with light are ones that had their tumors shrink in size, or they had, let's say, slower development, and they didn't develop as fast as their controls. But what I wanted to show you is that in the same time that we were working on these probes, we were asking ourselves, what happens to cathepsin activity in atherosclerosis? So in this project, we were actually collecting human plaques, so plaques that were taken out of the carotid artery of patients, and we were analyzing them by the severity of the plaque. So we had a pathologist grade these plaques, and we saw that they could be graded into 1, 2, and 3. And then we monitored using our fluorescent probes. As I said, these are covalently modified, so they attach to the cathepsins, and you could resolve this on gel and just scan the gel for fluorescence. This is an inhibitor treatment of these plaques, and these are non-inhibitor treatment plaques, and you could see how they nicely fluoresce. These are the bands of the cathepsins, and you see how they intensify as the plaque gets into a higher grade. We saw that, we quantified that. That was very nice. And then we could also see that asymptomatic patients that usually have the plaques at a lower grade have lower cathepsin activity, both of cathepsin B and of cathepsin S. That was cool. And then what we wanted to do is actually combine these two. We took animal model of LDL receptor knockout mice. So these mice tend to generate atherosclerosis in the heart and in the aorta. And we fed them with high-fat diet or with normal chow. So normal chow is here on the top. And these are our, as I said, these are hairy mice. So we have to shave them before we do this fluorescent imaging. This is done in the IVIS, in vivo imaging. And when we inject these compounds into normal chow fed mice, both of these are LDL receptor knockouts. We see that a little bit just accumulates in the heart. And this is a picture of the heart taken out ex vivo. But when we do this in high-fat diet, we see how nicely these probes accumulate right here in the aorta and the heart. When we take that out, we see that. And if we quantify the non-invasive imaging quantification, you see a significant increase. But the nice thing was that we tried to treat these mice when they were intact. So we just took a light at the right wavelength, which is near IR, really unharmful at 760 nanometers, and we shone light at these lesions of the mice, and then we extracted these tissues and we started to monitor them. I'm sorry for my mouse, it keeps on bouncing here. We monitored these lesions outside ex vivo, and we could nicely detect the reduction in the amount of macrophages in these tissues. So basically, this is a way that we could actually target the therapy of photosensitizers into the tumor, or into the atherosclerotic plaque. So after we did that, we were very excited. We could shift on to our CT probes. Here we were trying to work on a sort of smart probes, but that didn't really work very well. But we did have the basics of our just basic probe that covalently modifies these enzymes, and we wanted to use that for cancer imaging. So usually CT is used for anatomical imaging, and you could see that very nicely. And the reason that goes on is because the CT itself has very low sensitivity to different contrasts. You really need a lot of the contrast agent to accumulate in order to see a little bit change in the contrast of your sample. And that's why it's not really used for molecular imaging. But remember that our technology has this covalent modification that actually enables these compounds to accumulate in high levels. So we use this technology to try and generate a new series of probes. What we did there, the two very talented students there, Daria and Hanman, that I just showed their pictures, they had a whole set of systems. One was gold nanoparticles as the contrast agents for CT, and we had a very large series of different amounts of targeting and different lengths of pegs that were coating the nanoparticles. We had different sizes of the known gold nanoparticles, and we also had a control that lacked this targeting moiety altogether. And all, this is how it really looked. We have like this very short peptide that's recognized by the cysteine proteases, the cathepsins. You have the warhead right here, and you had attachment to the gold nanoparticle. We evaluated that these actually bind the enzyme. So we were worried that this big nanoparticle with all this PEG will cover up our little targeting moiety and we won't be able to see the inhibition, but indeed we see the inhibition. On the left, you see just a fluorescent probe without the gold nanoparticle or the probe. And then you could see increasing concentration of these different versions of the probes. And you see that they inhibit the activity, seeing a reduction in this fluorescent band. We could see the non-targeted probe. You could see how it doesn't do anything because it doesn't have anything that would inhibit these enzymes. Then we were very curious to know if these probes could actually enter into the cells because these are nanoparticles. So we did the series of different sizes of nanoparticles with different concentrations. I won't go very long into that, but indeed these nanoparticles inhibit the cathepsins inside the cells. And this is just incubating these nanoparticles in the growth media of these intact cells, and then looking at the activity of these enzymes. So we decided that the best one was version 4, and we then saw that it wasn't toxic and we could go in vivo. And we basically planted these tumors on the back of the mice, very similar to what I showed you before with fluorescent imaging or the photosensitizers. And we tested the different sizes of our probes into how they accumulate in the tumors. We had the non-targeted of the same size and the targeted compound of the same size. And what we're seeing here is a CT image of a micro CT of the animals that were treated with these probes when they were actually injected into the tail vein. And you can see very nicely how these probes actually generate contrast into these tumors. You could see them as you could see the bones. And you could see that the non-targeted don't really accumulate very well. They might accumulate just a little bit for the enhanced permeability and retention effect, the EPR effect. And when you could look at the cross sections, you could see this white rim and a little bit of inside the tumor itself, how this forms contrast. You could also see that the 100 nanometers doesn't really accumulate at all in the tumor region. We can quantify that, and I think this is actually pretty cool because what we did is we followed these tumors over time. So the tumor grows, and so we couldn't just look at the contrast. We had to look at the percent of pixels that had contrast for gold versus the total pixels that the tumor had. And we saw that about 10% of the pixels actually had gold. So 10% of our tumor had gold accumulating inside. And you can see the nice side distribution as the 100 nanometers didn't accumulate and the 10 nanometers did very nicely. And that followed very nicely when we extracted the tumors and just looked at the amount of gold using ICP-MS. We saw a very similar dose response. We also did a few controls here. We looked into the targeted nanoparticle. We had the non-targeted nanoparticle. We had a non-specific, what we call a scramble peptide, which wouldn't be recognized by our enzymes. So that didn't really accumulate. And we also had pre-treatment of our tumors with, or our mice actually were treated with the inhibitor when we injected it into IP, into the peritoneum. And we saw that that inhibited the activity. Thus, the probes did not bind and did not accumulate in the tumor and didn't form any contrast. And you see that quantification here on the bottom. So that was nice and significant. But I think the really most coolest thing that we actually saw is when we took these tumors outside of the mouse and we subjected them to electron microscopy. So we put them into the TEM. What we see here is one cell out of a tumor. And the tumor was actually a mouse that was injected with our gold nanoparticles into the tail vein. So the compounds circulate in the mouse, it accumulates in the tumor region, and it actually gets uptake into the lysosomes of these cells. So this is one cell, this is the nucleus of the cell. These are the lysosomes of the cells where the cathepsins actually reside inside, and you could actually see these nanoparticles accumulate. So these are the 30 nanometer gold nanoparticles in there, and they have the right size. So I think that was really proof that these compounds really accumulate right there. So in this project, we are now working into seeing the applicability to human patients. We're now trying to figure out what would be the best tumor or the best indication that we want to work with. We're heading into diseases that really have high mortality and lower detection limit. We're investigating colorectal cancer. We're investigating lung cancer. We have all kinds of things that I'm not going to show the data here, still unpublished. So I'm sorry about that, but we're very excited about that. So I'd like to conclude and say that we basically have a chemical technology to generate covalent modifications. Oops, sorry, that's my time to stop. I'll just conclude and say that we have covalent modifications that enable accumulation of our probes, and we could actually use these to detect our diseases. We're now shifting into ultrasound. So if anybody here in the audience is working with ultrasound and with contrast agents for ultrasound, that would be cool. We use these reagents not only for in vivo, we also investigate the involvement of cathepsins in different processes within the cells. If we have a few publications of the biological activity of cathepsins using these probes. And as I was saying, that this CT contrast will actually have a maybe clinical use in the future. So this is my group and the funding on the bottom. I'd like to thank you guys for listening. Thanks.
Great, thank you very much. Such an exciting talk, wonderful. Okay, we'll move along with Professor Aaron Wheeler. Aaron, are you here? Yes, I see. I don't see you yet, but I see that you're here. So Aaron, you can go ahead and upload your presentation. Meanwhile, just a few words about Professor Wheeler, also at University of Toronto. He's has a microfluidics lab. He works on developing lab on a chip for high throughput proteomics and drug discovery. And he's a very busy professor. So I asked him for some kind of a fun fact. So I can say that he's, first of all, he's now editor-in-chief of Lab on a Chip, the journal. So I'm sure that takes a lot of time and effort. And he has to figure out how to get some peace of mind when working at home with a small child. And yeah, with that, Aaron, please go ahead.
Thank you so much. Eylon, can I confirm that you can see my slides?
Yes.
Yes.
All is well.
Okay, so I'm very happy to participate in this symposium. The talks have been very, very interesting. I'm going to talk a little bit about our work with microfluidics, and I thought that since that topic hasn't been broached yet, I would give just a bit of background information. Microfluidics is the study and application of fluid flow in devices that bear micron-sized features. You're perhaps familiar with the kind of traditional mechanism of microfluidics, which is essentially tubes. They're enclosed channels or tubes on a planar substrate, and then one can flow fluids as streams or as droplets or many other creative mechanisms of moving fluids through microchannels. Another common motif is sometimes referred to as paper microfluidics. In paper microfluidics, fluid actually flows through solids. They're absorptive solids. The fluids flow by capillary action. And of course, this idea has been around for a long time in the form of lateral flow assays. A third format that I would describe is called digital microfluidics. And digital microfluidics shares some similarities, but also has some differences. I'll describe those in a few minutes. But the critical difference is that there are no channels and there are no dedicated paths. Droplets are just manipulated on an open surface. For any of these techniques, there's been a lot of interest over the past couple of decades about how we can use microfluidics to shrink the lab. And that's the vision has always been, I think, to shrink everything in the lab, not just the sensor, but also the sample processing, the various steps required to interface with detectors and so on and so forth. And this idea has been a very popular one that's really driven the community for, as I say, a few decades. The particular technique that I'll talk about today is digital microfluidics. This is a movie. I hope you can see it playing. Typically, the droplets on our devices aren't blue, pink, and yellow, and typically they don't spell U of T, but I think this is a nice demonstration of what we're doing. We're manipulating small packets of fluid on an array of electrodes. Let's take a look at this type of device in a little bit more detail. So here's a droplet on top of this array of electrodes. And what's important to notice is the droplet is not touching the electrodes. There's actually a hydrophobic coating between the droplet and the rest of the device. And in fact, we use something like Teflon as this coating. So the devices end up being quite chemically inert. If we look at a side view of the device, we'll see that in fact, in nearly all the examples I'll show you today, there are two plates. There's a top plate and a bottom plate. We have electrodes on both of these plates. Typically, the electrode on the top plate is transparent. So the pictures and movies I'll show you are peering through this transparent top plate. The other point I'll make is that there is a dielectric layer. And in fact, the dielectric layer is what drives the whole process. The dielectric layer allows us to treat each individual electrode on the bottom plate as kind of its own mini capacitor. And in fact, it's quite easy to estimate the forces and thus the velocities of droplets moving on the surface simply by accounting for the polarization of the dielectric and the energy that's then stored in that little capacitor. And then this picture here with the water in the comb reminds us that using stored charge to manipulate the position of water droplets is nothing new. It's something that we're all familiar with. Okay, so why would we consider using this technique? Well, it is quite versatile. It works with biological fluids. It works with aqueous buffers. It works with surfactants. It works with organic solvents. It's also a very powerful tool for sample processing. And if this video is playing for you, you should be able to see a droplet containing a suspension of magnetic particles. And this is a pretty nice format, because then if we bring in a magnet, use the magnetic field to pull the particles out of solution, we can then later come back in a second step, resuspend the particles in a new solution and affect buffer exchange. And you can imagine how nesting together a number of these sequential steps could allow for quite complicated sample processing regimens. The technique is also very well suited for reactions, and we've done a fair amount of work with some organic synthesis colleagues developing nanoscale reaction platforms in which we do split pool synthesis. And then finally, the technique certainly can interface with lots of different detectors. I'll emphasize some work. In fact, this is an artist rendering here of a chemiluminescent detector, but we can integrate with lots of other detectors. But I don't think I've told you anything unique yet. And in fact, in the literature, we could go find lovely examples of using microchannels or other kinds of microfluidics to do all of these things. The thing I want to emphasize here is these are all the same device. I chose these examples because they're all the exact same device. This is a generic device architecture, and this is perhaps a subtle idea, but it's a very powerful idea, that you're not building the process into the microfluidics. This allows us to essentially take this one generic platform and build up a menu of different processes. Here's some processes. On the top left, we see kind of a graphical user interface of how we've programmed the droplets to move. In the top right, there's a movie of droplets. By the way, our droplets don't always contain dyes. We typically put the dyes in the droplets to make them more visible. And then a couple of other sequences below. We can build up a menu of these sequences, and then we can call them. And not only can we program repeated calls to different programs, we can actually decide on the fly, which again is quite powerful. I want to emphasize one more time how unique this is. We have a lot of flexibility in the real lab. When we walk into the lab, we could decide to dilute and then dilute again and then dilute again. But in a conventional microfluidic device, you have a built-in dilution module. And if you discover in the field, for example, that you need more dilution, you're in trouble. Whereas with this digital microfluidic technique, you have the flexibility to make those changes depending on what you want. One other point I'll make is that this really is on a chip. And by that, I mean, we're not mating this chip to tubes and interconnects and valves and pumps. That's not to say those things are bad. In fact, I really love those things. They're very useful. But in its purest form, this is just a solid state device with no moving parts. And that has potentially some advantages. Okay, so my group and I have spent a lot of time over the years exploring potential applications for digital microfluidics. One area that we're spending a lot of time these days is building miniaturized cell culture and analysis systems. And in particular, we're very focused right now on doing some single cell analysis where we grow up arrays of adherent cells or ex vivo tissue sections, and then we come in with a laser and we pick individual cells. We pull those droplets off to do transcriptomics or proteomics or genomics. Another area we've spent a lot of time working in is simply using this technique as a sample processing module. So let's take a complicated sample like a tissue section or a dried blood spot on a piece of paper, and let's build a system that allows us to do all of the sample processing steps that are needed to analyze these types of samples. It ends up being a quite convenient package for these kinds of applications. I'll point out that for these kinds of applications, we very regularly use large instruments, and I think this is a fine model to take. It's not a model that works out in the field, but it works great in the lab. So for example, we've built an interface that we have used and used and used, where we have the auto sampler on a little HPLC pump come in, pick up the droplet and inject it onto a column and then do mass spec. We've also done some really crazy experiments where we've taken our devices and jammed them down inside the bore of the magnet in an NMR instrument, and have done experiments down in the NMR, and that's been a lot of fun. But today I want to talk about taking our instruments outside of the lab, and Keith gave a very nice introduction of this idea and why this idea is important. There are lots of places where we don't have access to the lab. So is it possible to take a miniaturized lab to those places to do the diagnostics there? And this work has arose from just a wonderful collaboration we've had from the US Centers for Disease Control. Several years ago now, they approached our lab and said, hey, we know that you have developed some nice diagnostics for measles and rubella. Could we take this out in the field? And specifically, could we take this to a refugee camp? Our problem is we have a refugee camp where we don't know the immunization records of the population. We really need to know who's immune and who isn't immune so that we can figure out when to do vaccinations. And of course, this was kind of a new idea to me back then. These days, during the global pandemic, I think we're all familiar with wanting to know this type of information. Well, of course, you know, the traditional way to do this is we would collect samples from patients. And in Toronto, this is exactly how you do serological surveillance. You go to the doctor draws blood, the blood is sent to the lab, and a few days, maybe a week later, you get the information about your titer for that particular immune response. That doesn't work in a refugee camp where there isn't a centralized lab. And often we're looking at months, if ever, that you can get the results back. By that time, the population of the refugee camp has completely changed. So the idea then is, again, this lab on a chip idea. Can we take our instruments into these very remote settings and get laboratory quality results? And that's what we set out to do. I'll point out that until this point, we had never taken our instruments outside of the lab, and it proves to be a really big challenge. And in fact, I'm going to tell you some success stories, but the stories aren't always successful. It's decently straightforward to make an assay work in your laboratory. It's a real challenge to make it work out in the field. Okay, so what were we doing? The actual assay itself was quite straightforward. It was just a simple sandwich style ELISA. In this case, we're taking magnetic particles that are coated with inactive virus, and what we're fishing for, our analyte, is the antibody. And we've done both IgG, we've done IgM, we've done IgA. In this particular case, we were looking for IgG to tell us kind of long-lasting immune responses. We mix those particles then with a secondary antibody conjugated to an enzyme. And of course, we've selected an enzyme that will return a chemiluminescence response. So here are a few pictures. This is a picture of diluted blood. Again, I've added some dyes here just to make the droplets more obvious. And then at the end of the assay, we take the droplet with the chemiluminescence and we make the measurement. The other big challenge we had is doing a few experiments in the lab. I can afford to make very expensive devices, but doing hundreds of experiments out in the field, I just don't have the funding to build fancy clean room devices. So we've spent a lot of time developing techniques to build really inexpensive microfluidic devices. So our best techniques these days are built using inkjet printing onto rolls of flexible plastic, and then taking those rolls of flexible plastic and doing some roll coating processes. This ends up being a really inexpensive way to make workable devices, and we're able to assemble these devices for less than $1. The other thing we had to think about was how rugged our instruments were. And again, ruggedness is really important for this application, just because the conditions out in the field are poorly controlled. So we spent a lot of time prototyping our instrument. This is an old video of an old prototype, but it's a time-lapse video showing one of the undergrads in the group assembling this early version of the instrument. And our goal was to get the instrument running, to knock it off the table, and then pick it back up and have it still able to keep running. And we got to where it worked most of the time under that condition. Okay, so after solving some of these technical challenges, we took the instrument out in the field with the Centers for Disease Control. And in particular, we went to Kakuma refugee camp. It's up in the northwest of Kenya, close to South Sudan. A lot of the population is coming from South Sudan. And of course, there's no vaccination history to speak of, and there's just endemic outbreaks of measles and rubella. Over the course of a few weeks, the team ran nearly 300 assays from about half as many subjects. These were primarily mothers and their children, and it coincided with a vaccination campaign. So in some sets of subjects, we were able to test them before and after vaccination, and those were particularly useful samples. We found the flexibility of this system to be quite critical because we faced several severe challenges, one being one of our reagents lost potency. At this point, we were working with liquid reagents that had to be to remain chilled. And we think one of them warmed up at a certain point in the many plane connections that were required to get to the site. So being able to use larger amounts of that reagent and to incubate for longer duration was critical. Without this flexibility, we wouldn't have made this thing work. The other flexibility that proved really useful was the capacity to run different combinations of tests. Depending on who was coming in and getting which vaccination. It could be measles or rubella or both. So having that flexibility was nice. The results were quite good. If we look at our receiver operator characteristics, we have an area under curve of 0.9 for our rubella test in this example, and an area under curve of 0.86 for measles in this case. Again, in this particular test, we were running IgG assays. We've since then gone on to do many other kinds of immune responses. Let's see, why are there differences? Well, it's kind of amazing that they're so similar to me. These tests are very different. We're working with whole blood. The reference test was serum after freezing and thawing. We're using different antibodies, different substrates, different volumes. So the fact that they're pretty close, and I have 2 minutes, so I'm gonna quickly run through. I'll say that we've done another test, an even bigger test. The really exciting thing for us is our instruments seem to work quite well. We are, believe it or not, still waiting for laboratory results. This COVID situation, as you might imagine, really has occupied the CDC's attention for the last year and a half. So we hope to get back and finish this story soon. At any rate, our instruments work in the field, battery powered, 10 hours a day. They seem to work quite nicely. We're doing a little bit of work with COVID these days as well. So let me conclude by saying I've introduced digital microfluidics. If you're not familiar with it, I think it's a decent tool to take out in the field. I also think it's just a useful platform technology. If you can think of an application that it might be useful for in your work, please come talk to me, or please forge out and use this technique. There are now a couple of little companies that have instruments for sale. I happen to know a little bit more about this one, but there are others. I think it's a technology whose time has come, and I encourage you to give it a shot. With that, let me thank the research group. You know, it's fine to see you guys through the computer these days. That's kind of been a nice thing that's emerged, is how easy it is to connect with colleagues. But I really hate that I'm connecting with my own group these days only via these intermediate means. I'm really looking forward to seeing these guys in person. They are just a joy to work with. I'll thank these organizations for funding, and I'll thank you for your attention.
Great, thank you so much, Aaron, also for being on time. So I just have just a note, or I'm wondering, are your labs closed in Toronto?
Our labs are open, but there are strict limits on the density. In fact,
okay.
Irrelevant people like professors are usually
okay.
I came
see.
in today
Okay.
for the stable internet.
Okay. Got it. Okay, thanks so much, Aaron. It's a really cool talk. I guess we can move along to our next speaker, Professor Katy Margulis. She's actually a new recruit at the School of Pharmacy at the Hebrew University. So we're very happy that we have Katy with us. Katy works on the design of novel therapeutics and drug delivery systems using nanotechnology and mass spec imaging. And her fun fact is that she loves skiing, but she actually didn't choose the best country for it. Although we have once a little bit in the winter time, we have the Golan Heights. We have a mountain which is 2,000, a bit over 2,000 meters high. So you're Canadians, you know what a meter is, right? So it's 2,000 something meters high, and we have a bit of snow there. Yeah, I mean, Europe is not so far away, so it's not too bad. Katy, can you, does your...
Yeah, I'm trying to share my
Can
screen.
you...
Yeah.
Okay.
Just a second.
Sure.
I was listening to you and
Okay.
just wanted to tell that actually skiing allows me traveling to other countries.
That's
but
right.
I haven't skied
That's
for
right.
3 years now, so
Right.
maybe next year in Canada, who knows?
Yeah.
Okay, so let me just...
And Katy's, the title of her talk is Mass Spectrometry Imaging and Therapeutics, unless it has been changed, but we see, yeah.
Yeah. So
It's
Just
there.
a second.
It's there.
It's there. They're coming
Yeah.
there. Okay. So can you hear me? Can you see this screen?
Yes. All's well. Yeah.
Okay, so it's a great pleasure to be here today, and thank you so much for inviting me in your wonderful talks, and it's really inspiring. So let me share some of my data on mass spectrometry imaging for therapeutics. I have to say that I'm quite aware that University of Toronto conducts a very impressive research in mass spectrometry imaging field. So it's definitely not a new topic for you, but I wanted to bring in other aspects and show you how we can use those imaging techniques to discover new therapeutics and maybe predict the sensitivity of different diseases to treatment. And so mass spectrometry imaging can be sort of imagined as a high resolution molecular microscopy. You just take a slice of tissue, of human tissue or animal tissue, and you scan it. And after you finished, you have distributions of hundreds to thousands molecular ions in the tissue. You don't need to know what you're looking for. You can just scan and get plenty of new information. The technique is pretty fast and sensitive, and nowadays we can get to pretty good spatial resolution with this imaging. And so mass spectrometry imaging is based on mass spectrometry. This is our raw data, so we get mass spectrum. In this mass spectrum, you can see all those peaks, which actually each peak represents a different molecular ion. And the x-axis is mass to charge ratio, which actually is pretty unique to each and every ion. And the y-axis is the relative abundance of each peaks relative to other peaks in the spectrum. And so even by glancing at the spectrum, you can see that we have hundreds to thousands ions in the spectrum. And if we zoom in, we can see that there are much more. So the information that we're getting from the single scan is huge. And so how do we know the molecular structure, the chemical structure of each molecular ion? If you have a good mass spectrometer with a decent mass accuracy and a resolving power, even by looking at mass to charge ratio, you can already identify your molecular ions with pretty good confidence. But we can also fragment those ions to little pieces, identify each of those pieces, and then put them all together to assemble a parent ion. And then we are pretty much sure about chemical identity. So this is mass spectrometry, how we get it to imaging. Imagine a tissue slice with different pathological or normal regions. And if we are able to extract analytes from a single point on this tissue, we can get a mass spectrum. Now, if we scan this tissue in two-dimensional manner and we have many of such points, we will get many of spectra. And so each and every peak of our spectrum can eventually give us the distribution of the molecular ions throughout the tissue. And those are heat maps, which means that if the color is warmer and brighter, it means that the relative abundance of this peak and this point is higher. But if the color is colder, then we have a lower abundance of this molecular ion in this specific region. And so our favorite modality for mass spectrometry imaging in the lab is desorption electrospray ionization mass spectrometry imaging, because it's fast and simple. So we put, our tissue slice on the two-dimensional moving stage in front of mass spectrometer. We spray the tissue with the spray of charged solvent droplets to desorb and ionize molecules from each and every point. Then the splash of those droplets take the analytes to mass spectrometer, and then we move this tissue in two-dimensional manner. And at the end, we can get the distributions of molecular ions in the tissue. See, it's very fast and very fun to work with. And now let me tell you how we use this technique to discover new therapies. This is the project that I started during my postdoc at Stanford University with Professor Dean Felsher, who is an oncologist at Stanford, and we still continue it. And so Professor Felsher's lab knows how to create conditional transgenic mice models that will express MYC oncogene. MYC is a very known human oncogene that when mutated will cause a variety of human cancers. So Professor Felsher knows how to create mice that on demand he can turn MYC oncogene on and create cancers in certain organs. And then he can turn MYC off again, and then the cancers will regress. So this system is very interesting for our studies because we can study each and every step of cancer formation, progression, and then regression. And so they know how to form cancers in different organs and how they actually do it. They take a truncated MYC oncogene, and then they couple it with organ-specific promoters. So the cancer will be formed in only one organ. And then they couple the whole thing with inducible unit that can only work dependent on the presence of tetracycline family antibiotic around it. And so those mice, from the day they're born, they receive doxycycline, which is tetracycline family antibiotic, in their drinking water. And then they're completely healthy. They will not transcribe the whole sequence. But when you remove doxycycline from their drinking water, they will start forming tumors in this specific organ that we are interested in. And so then we come and then we study the formation and regression of tumors. And so we studied many tumors by DESI, but let me show you the work that we've done on renal cell carcinoma or kidney tumors. So in those mice, we activated MYC for 15 days, and then we switched it off for 10 more days. And at each point, we imaged the kidney with a mass spectrometry imaging. And so we were interested in metabolic signature of tumors and to find out how MYC drives tumorigenesis and maybe find some options for pharmacological intervention for new therapies. And so this is our raw data, average spectra from control kidney of mice that did not induce, they weren't, where the MYC oncogene wasn't activated, then with MYC on kidneys, and then finally with MYC off after oncogene activation. So even with the naked eye, you can see that the spectra are completely different, although control and MYC off seem kind of similar in these higher molecular weights, but MYC on is completely different. And one of the most prominent players here, new peaks here, are those two guys. And if we look at their structure, they're actually phosphatidylglycerols, phospholipids or that have glycerol moiety substituent on their phosphate group. And so not only the two of them, but many more, around 20 molecules were identified as dramatically changed in MYC on activation. And all those species were highly upregulated in MYC-1 activation in a renal cell carcinoma. But not only in renal cell carcinoma, then we looked at other tumors which were MYC dependent, and all of them, phosphatidylglycerols were upregulated. And so it made us look at the phosphatidylglycerol synthesis pathway, and we saw that almost every enzyme that we checked on this pathway, it was upregulated by MYC activation. So MYC drives tremendously towards a phosphatidylglycerol synthesis. And so we thought that, okay, let's inhibit one of these enzymes, specifically glycerol phosphate acyltransferase, and let's see if we decrease the amount of phosphatidylglycerols that a tumor can synthesize, how it will react to this change. And so we took a known inhibitor of this enzyme, which was never tried for cancer before. It was trialed for obesity in humans, and it was found very safe. And so we gave it to our mice. So we gave only one injection to mice after 2 weeks of MYC induction, of MYC activation, and we saw a dramatic decrease in kidney weight. Kidney weight corresponds to tumor weight, practically. So the kidney weight went back to the weight of normal kidney. And this is untreated control after MYC activation. So the effect was just dramatic, and it repeated itself in other tumors. I'm not showing here the data, but it's pretty impressive. So mass spectrometry imaging just helped us identify a certain druggable target in MYC-induced tumors. And so another project that we started and we are still working on is actually how we can predict using mass spectrometry imaging sensitivity or susceptibility of diseases toward pharmacological intervention. And here we also worked with renal cell carcinoma, but human renal cell carcinoma. And this is a disease that currently doesn't have an efficient treatment. But one of the most prominent drugs for this disease is glutaminase inhibitors. Glutaminase inhibitors inhibit the conversion of glutamine to glutamate inside the cells. Now, it's known that human RCC heavily dependent on glutamine rather than on glucose for its metabolism. So if we actually inhibit this step and prevent glutamine from going to Krebs cycle as glutamate, then we can maybe make those tumors starve to death. So this was our hope. And so we used a new molecule, which is called CB839, CB839, and it's an oral therapy, oral glutaminase inhibitor. It is now extensively trialed in human trials, but when we started working on it, it only had the result of very small trial in humans. So 19 patients, RCC patients, received this molecule, and only 9 were responsive. And so the researchers couldn't find any predictive features, either tumor type or immune system involvement or genetic factors that would predict the susceptibility of those tumors to this treatment. And we thought that maybe by looking at metabolic distribution in the tumors, in tumor biopsies, we can predict the susceptibility. And so we took 2 human tumors and then transplanted it into 21 mice. And then we treated those mice either with this glutaminase inhibitor or placebo orally twice daily. And then we checked the distribution of metabolites after 3, 7, and 21 days. And our goal was to predict early on during treatment, when tumors do not show any other signs of a responsiveness just by looking at metabolic analysis that the tumor will respond eventually to treatment. So this is what we hoped for, to take two healthy looking tumors and say, okay, this one is responding and this one is not responding. So here you should switch to another therapy, and here you can continue with this one. And so we first imaged the kidney after 21 days of treatment, and we identified 44 mass spectral peaks that were they were predictive of a tumor response. We ran a machine learning algorithm called a gradient boosting decision tree algorithm to identify those peaks. And after 21 days, 3 weeks of treatment, we can definitely say, okay, based on metabolic distribution, we can say with 98% of accuracy whether the tissue was responsive or not. And then we ran those predictive algorithms on 3-day treatment tumors. And by the way, after 3-day treatment, our pathologist looking at the histopathological staining could not recognize any predictive feature of tumor responsiveness, neither when they looked at KY67 staining, which is predictive of tumor proliferation, all look similar. But using our metabolic imaging, we could already identify with 80% pixel by pixel data the responsiveness, and on the tissue basis, this accuracy went up to 100%. So just by running our mass spectrometry imaging, we could with 100% accuracy say, okay, this tissue is going to respond eventually to treatment. And so you might ask, okay, this is a glutaminase inhibitor. Why can't you just look at the ratio of glutamate to glutamine and see if it's working? And surprisingly, this was not predictive of treatment success. So after 3 weeks, there was some accuracy of prediction, but definitely not after 3 days. We almost didn't see any difference. So we actually need to use the whole bunch of peaks that we discovered in mass spectrometry imaging to see these predictive features. And with that, I think my time is up. And I want to thank you all for your attention. And this is my newly built lab. This is my team. And we hope to grow more in the coming year. And those are the funding agencies that helped me build my lab. And thank you all. Thank you for having me.
Great. Thank you very much, Katy. Very nice talk, and thank you for being on time. As I mentioned, we'll have questions at the end. Okay, so we can move on to our last speaker of this session today. And I'm happy to present Professor Warren Chan. Professor Chan is the head of the Institute of Biomaterials and Biomedical Engineering at University of Toronto. Okay, so I see that you're putting up your slide. That's great. Professor Chan works on the development of nanoparticles for cancer targeting and infectious disease diagnostics. Warren, can you bring up your slides? Is it working? I don't see. Warren, are you with us? Okay, great. So just
are the slides up?
Yes, they are, but you could do it at a presentation mode. And just one fun fact about Warren, I think, is that he's finally, with the COVID-19, he's starting to appreciate the concept of sleeping. So I don't know, maybe he didn't sleep before COVID-19. So anyhow, and his, I think, is there a title there, Warren?
I did not put a title.
Okay, I can say the title. It's okay. It's cool. Don't worry about it. The title of his talk is COVID-19 Diagnostics via Quantum Dot Barcodes. So Warren, all yours.
Thank you very much. Yeah, one of the reasons I didn't want to put a title up at the beginning, right, is because I want to actually put an emphasis in terms of the lab members in my lab that have been really pushing toward developing some of the diagnostics for COVID-19. So this presentation will focus on their work, and they've been really pushing hard since probably about March of last year to develop these technologies and to translate these technologies. And I just want to start off by acknowledging Matt, Aiden, Johnny, Hannah, Vanessa, Bodisha, Provnov, and Hong Min, who have been really key to some of our developments. This particular project was funded by the University of Toronto through the Connaught ISI program, as well as the CHR and NSERC CHIRP program. I also like to acknowledge our collaborators, Sam, John, Allison, and Gary. So I've been working with them for the last probably about 5 to 10 years in terms of developing diagnostics for infectious disease detection. So when COVID hit, we all jumped right onto that particular application. So Sam's at Sunnybrook, Allison's at State Kids, John's at a public health lab, and Gary's in CCBR. And they're researchers who have been very key to what we've been working on for the last bunch of years, or last actually year. So I'd like to start off by discussing the central concept of our diagnostic tests. Really, our objective is to develop home serological diagnostic testing for monitoring infected patients. It's actually very easy to design diagnostics for a nice lab and for a nice clinical lab, but it's very challenging to be able to translate those particular technologies for home use. So the overall idea is pretty straightforward. I think Aaron already mentioned it in his seminar. So you have blood collection, and then once you collect your blood, you essentially have some sort of a serological test or genetic test to be able to measure certain biomarkers in the blood samples. Then you actually use a smartphone system to essentially detect what's been tested. And what the smartphone does is two things. It allows you to relay data to a centralized facility, but a smartphone also may potentially allow you to analyze the data. So it allows you to actually globally map what's going on, and it allows access by the general public in terms of the positive, negative results, or how positive, how negative some results are. And so this whole scheme has been developed now and very mature actually in some sense, because a lot of other people and a lot of other countries are actually developed the same kind of technology. So central to our diagnostics is a concept called quantum dot barcode assays. So the concept is pretty straightforward. You have polymeric beads, and essentially you infuse different color fluorescent quantum dots inside the beads. So you have a green bead, a yellow bead, a red bead, a blue bead, and you can actually put different combinations of barcodes in them, quantum dots in them. So this one would have a blue and a green, a yellow and a green, and so on. So you can actually go up to 5 different colors. So each of these particular beads now becomes unique because they have a unique optical signature. And so this is equivalent to what you see in a grocery store when you have little lines and a number that's underneath it, because those particular numbers identifies the product that you're actually purchasing. And the reason we use quantum dots is for two major reasons. The first one is that you can actually design different emitting quantum dots by changing the size of the nanoparticles. So something that's small, that's 2 nanometers, would emit blue light. Something that's large would emit red light, and all the other colors would come in between. You can also design the quantum dots to emit in the blue range by changing the composition. So going from cadmium selenide to cadmium sulfide, or you can change it to the near infrared by using mercury sulfide, indium phosphate, and so on. So the quantum dots allows you to tune the light emission by changing these particular parameters. The other thing that's very important for this particular applications is that the quantum dots tend to be very photostable, about 100 to 200 times more photostable than organic fluorophores. And so that's very important because if you're trying to read the optical signal of the beads, if they lose the signal, that actually allows you to misidentify that particular bead, right? So the more stable it is, the better it is in terms of detection. And the other advantage, excuse me, is that you can actually excite all the different emissions of the quantum dots by using a single light source, a single wavelength. And this is very important because it starts to simplify the instrumentation design in terms of identifying the optical signal. So the way we use these barcodes are the following. So you have a polymer beads. Inside them, for example, you have a green fluorescent quantum dots. You conjugate a recognition molecule on it, on the surface. So the the green signal recognizes the molecule you put on the surface. So now all of a sudden you have a yellow bead, and the yellow bead now is recognized by a different protein. So protein 1, protein 2. So the bead color identifies those particular recognition molecules. So if you can make 1 million different barcodes, you can actually detect 1 million different types of recognition molecules that are coded on the surface. The detection process occurs when you actually have a secondary structure. For example, you have a bead here. You have, let's say, an antigen you coat on the surface. So the color of the bead essentially identifies the antigen you place on the surface. Then if you're trying to measure the antibody essentially now binds onto the antigen on the surface. And then what happens now is if you incubate it with another antibody with a secondary probe, which is indicated here by the little red dot on top, what you see now is a sandwich structure. And so when you optically read your signal, here you get a green signal and a red signal. The green signal identifies the antigen you place on the surface. The red signal tells you whether that antibody is present or not in your biological fluid. And so if you have a multiple different fluorophore encoded beads, you can detect multiple different types of antigen. A negative detection occurs when you have a bead, you place an antigen on the surface, there's no antibody that recognizes that antigen, and all of a sudden now your secondary probe does not stick onto the surface. And when it does not stick onto the surface, what ends up happening is that all you now read is a yellow bead signal, right? And because you just read a yellow bead signal, that tells you you don't have a detection for that particular antigen in your system. And again, the nice thing about our assay is that you can actually design, for example, 4 different fluorescent emitting quantum dot barcodes. You can add it all into a centrifuge tube for reaction along with your secondary probe. So you mix it up and then you form the sandwich structure. And so you can do hundreds and thousands of different barcode system in a single reaction file. Again, when you excite all the signal, a positive detection, you expect both the beads as well as the secondary probe. And that's how you essentially do the detection. And so we first designed these codes, these barcodes for 2 types of antibody against one against the nucleocapsid and one against the S1 receptor binding domain of the SARS-CoV-2 virus. And then we also have negative and positive control in terms of our experiment. And that validates that the experiment works properly. And what we see here, right? So the first test we did in terms of our bead was do a direct comparison with ELISA. So ELISA is essentially the gold standard in terms of measurement. And what we see is that our assay, again, it can detect about 10 to the minus 11 molar of the antibody against these particular antigens. And if you do ELISA, it's actually very similar. So when we do ELISA as a head-to-head comparison, we're also getting around the 10 to the minus 11 to 10 to the minus 12 range, right? So our assay is very similar to ELISA, but the difference of our assay is that we can multiplex. And I'll show later on how we can actually make this as a portable serological test at the end. And then we also did a clinical performance analysis for the antibody against nucleocapsid as well as S1 RBD by comparing it to ELISA confirmed positive results. And we will show that with the nucleocapsid, we are getting 89.5 sensitivity and a specificity of 100%. And similarly, the S1 RBD is about 95% and 100% in terms of sensitivity and specificity. So once we confirmed that our assay works relatively well with ELISA, our next step was then to start to build up a simple reader system. As I mentioned, one of the unique advantages of using quantum dots as a barcoding scheme is that the properties of quantum dots allows you to easily design the optical readout system. So you don't need multiple lasers, which you would need with multiple fluorophores. You can also, because their photostability and their brightness, you can also use a smartphone camera to be able to image single beads pretty easily on the surface. So in this assay, we actually designed a cartridge which essentially automates the assay. And one of the ways we automate the assay is that we actually put magnetic nanoparticles inside these beads along with the quantum dots. And then we can actually use a magnet to pull the beads to the side, wash, re-incubate, wash. And so you have to wash each step to remove non-specific binding. Once we essentially do the reaction, then we essentially inject them into a microfluidic chamber. And in the microfluidic chamber, we actually contain a electromagnet that's underneath. And what it does is it slows down the transport of the nanoparticles through the chamber. And once it slows down, it allows the smartphone camera to start to capture the image. And we've actually written an app to deconvolve the barcode signal, because in the app, we can identify what the signal, what the antigen or the molecule that we're trying to detect, as well as a secondary probe. And so with this device in place, with this device in place, we can actually then essentially did clinical testing and clinical performance with the smartphone system. And it's actually quite good. So in most cases, we're getting approximately 90% in terms of the clinical sensitivity and 100% specificity. So here's just an example of the results where the cutoff is, right? So the negative is where we misdiagnose when we are trying to detect the one type of target versus a different type of target. And so most of the signal samples we have were in the positive part, so they show a positive detection. One of the things that we noticed that when we start to use multiple beads, so if our strategy was that if one of the two beads produce a positive signal, we would say that's a positive detection. We're getting anywhere from 95 to 97% in terms of clinical sensitivity. So it's actually quite good in terms of detection performance. So once we actually have validated this device and system, we actually have built a broader scheme to be able to use our barcode system. So a patient would essentially input their information. You have the barcode detection process. And as I mentioned, this is the barcode signal. This is secondary probe signal. Once the result becomes positive, we actually created an app, a central database where the information can feed into this data set, right? And so this was created by Gary Bader. And you can see that we've coded in things like the age of the patient, the sex of the patient. We also coded whether the patient have different types of pre-existing conditions or is taking certain medication. And so you can actually build all this data in place to allow you to analyze it. And we can also see where it's locating at in terms of the Toronto area. So positive detection, you can see a certain number of cases. When there's no cases, you see empty spots. So this allows a real-time tracking of information in terms of the detection process. So as Aaron mentioned, serological tests are useful for monitoring immunity. So given the fact that there's a lot of vaccines that are now being administered globally, there's still questions about whether the immunity is retained from patients that has been vaccinated or actually been infected with SARS-CoV-2. So such a device could be very useful in terms of monitoring immunity. It's actually easier to do this at home than it's to do this in the hospital system, just because it would take more effort for people to go to a hospital versus at home, you just push a button and you get your results, and then it sends it to a central database. And the other thing that we would like to do then is to use this to monitor infection. One of the problems right now is that a lot of patients, when they get sick and they have certain types of symptoms, the symptoms for SARS-CoV-2 are similar to other symptoms, like flu is a good example, common cold. And so we actually now are developing different barcode schemes with different targets that can actually differentiate whether you have SARS-CoV-2 or other types of infection. So again, the multiplexing will allow you to do those type of analysis as compared to a single plex assay or system. And the other thing that we actually did is that if we're going to use for this particular test for a home-based test, we're comparing it to lateral flow assays. Right now, commercially, there's lots of lateral flow assays that's being sold at Shoppers, Walmart, and at your local pharmacies. The problem with lateral flow assays is that the analytical sensitivity is actually very poor. It's about 1 million times less than that of ELISA. So what it does is that it actually gives a lot of false negative results very quickly. And last year, during May and June, the US FDA approved a lot of lateral flow tests, and they actually created a lot of problem because there's a lot of misdiagnosis of patients. So aside from lateral flow, there's no other home-based tests at this particular point. And so again, if you look at like patients that have severe symptoms from SARS-CoV-2 infection, moderate and mild, so we compared these three patient population. And what you see here is that with a lateral flow test, they're not able to detect very well, even with the severe cases. So it misdiagnosed at least 50% of the cases. So in our system can detect 100% of those cases. When it's moderate symptoms, our test is anywhere from about 95% or so in terms of clinical sensitivity. But the lateral flow now drops to 40%. And if it's a milder case, it's less than 25% positive detection in terms of lateral flow versus our barcode. Our barcode is about 85% or so. So now imagine if you're asymptomatic and you're trying to measure certain antibodies in the bloodstream, lateral flow can't actually measure those very well at all. So it just misdiagnoses patients who are asymptomatic. Our barcode system may not be at 80%, but I'm guessing with asymptomatic cases, we're probably around the 70% or so using serological tests. And again, if you look at long-term monitoring of patients, so we did this experiment where we took patient samples at different days post-infection. So without diagnostic tests, it was pretty, we can detect 100% of the cases up to about 50 days. After that, there is a waning of antibody levels, and we can see some decreases along the way. But if you use the lateral flow test, you can measure for a certain amount of days, and then all of a sudden now you're kind of misdiagnosing past 45 or 50 days or so. And these are the ones that we took patients that we can detect very well at the beginning. So we took this in terms of both samples. And again, we're tracking those patients for long term. The barcodes works better than the lateral flow. And the real reason is because the analytical sensitivity is much better with the quantum barcode system, right? And again, we map this data in a different format, but you can clearly see that the lateral flow does not work after a certain day in terms of area efficiency. As compared to our system, it's much higher in the global scheme, and that's because of the performance of our system. So beyond serological, because we all know that a real positive test right now is a genomic test, not a protein test. A serological test usually tests after you've been infected past a certain amount of days, but we're really now building a genomic test for detection. And then genomic test, you need to go all the way from extraction to detection. And right now, there are very few techniques that goes from extraction to detection in terms of full automation, from getting the swab all the way to detection. A lot of cases, most of the diagnostic systems that's available for genomic testing, what they do is actually they extract out first, and then after they extract out, they basically then run the amplification and detection process in a second step. And so that actually can create human errors that slows down the process. So all these things start to limit in terms of those use, right? And so right now there's one or two devices, the BioFire and the Abbott, that has extractions involved, but there's still problems in terms of clinical sensitivity and specificity of those devices and systems. So in terms of the effort, this is actually one of the areas that needs to be integrated in the development of diagnostics, is to integrate all the steps of the process into a single useful and easy to do device that can be done for home care or home use, which is not that straightforward to do. But here's a device that we've created the last year. We're still optimizing some of the components, but it's not that far from finishing up at this particular point. So the other thing about home tests is that when we actually have multiple components for the barcodes, it's actually very easy for technicians and for people who are skilled in terms of doing this type of work. So a couple of years ago, we thought about ways to deliver the reagents that made life easier for everybody. And so what we started to develop in 2017 is a technique where we're essentially putting all the reagent components into a tablet. So if you have 3 steps, you may have 3 different colored tablets. And so you add that into your reaction, the tablet dissolves and it can bind to its target. So this makes everything easier, because if you're planning to use this for the home user, what you want to instruct them to do is add a green tablet, a blue tablet, and a red tablet, and then you read the results. That makes things much easier and more practical, right? And so we're starting to build this up in terms of the tablet system. We're starting to develop multi-layer tablets. So we're actually now developing tablets which have 4 or 5 layers with control release, so that you can just add 1 layer in terms of the reaction. And the device itself right now is automated. You just push a button and then you actually get a result at the end. So the conclusion that we have is that we've developed a series of multiplex COVID-19 diagnostic technologies. So the serological test is complete with clinical evaluation at this point. And we're now continuing to push, trying to finish up the genomic test device that allows us to do head-to-head comparisons with PCR and patient samples. We've also developed other types of diagnostic tests that are based on colorimetric. And so those studies are also in review at this particular point. And these tests allows you to look at the results with the naked eye, and you can actually differentiate. One of the papers we submitted allows you to differentiate between the flu and SARS-CoV-2 infection. And so we're starting to expand this to develop other respiratory panels because this is really a respiratory disease at the end. And so we need to differentiate SARS-CoV-2 from other respiratory disease. And the reason for that is because the treatment strategy is very different based on whether you have flu, cold, SARS-CoV-2, and it's important to understand at this particular point. The other steps that we're taking now is that we're starting to partner for translation. A company I started about 4 years ago, Luna Nanotech, so they'll be taking the lead in terms of commercialization. And we started discussion with a manufacturing company in Korea to start to scale up production of these particular device and systems. So the company's also been supplying a lot of extraction kits and reagents for the global community. So the money that the company made from selling SARS-CoV-2 diagnostic components are going to be used to pay for the manufacturing of the devices and reagents moving forward. With that presentation, thank you again to Brian for inviting me to give this seminar, and I'd be happy to answer any questions anyone may have in the session, in the next session.
Okay, great. Thank you, Warren. Thank you for such an interesting talk. Amazing stuff. Okay, so what I think we'll do now is I'll just read out the Q&As that have accumulated. There are not many, but then I think we can have like a few minutes of just like open discussion. If anyone wants to ask questions, then please feel free to do so. So I'll start with a few questions that were given to me. And I'm not sure, Laszlo, if I'm pronouncing the name correctly. But the, one question is there an ability to multiplex FIT-PNA using a longer nucleic acid sequence barcode? So we are a bit limited in terms of, I'm not sure if that's the question, but in terms of the length of the PNA, we can't make them too long because then they become kind of greasy and they kind of bind to each other or intramolecularly. So we're a bit limited there. So I would say maybe can make something as long as 20, 25 MERS. And the other question was if we have been tested on FFPE samples. So yes, we have done that in the past on frozen section and so on, and that works. But obviously we want to use this, you know, and as a very fast detection mode for biopsies, you know, not to wait weeks for it to get an answer, but just to get a quick answer by simple spraying. And the last question that I had here by Julia, so I hope she's still around, is that how do we decide on what biomarkers? That's a bit of a tricky question because usually we're a chemistry lab, so usually what we do is we ask our collaborators, the biologists, what's really burning in their, like what would they really like to detect. But we're not limited. I mean, we can make a PNA for any sequence, so the sky's the limit. Now there's a question here for Keith. So given the ultra sensitivity, could you comment on false positive? And are you contemplating using the system on ctDNA for cancer detection or recurrence detection in blood?
Okay, cool. So yeah, the sensitivity of the assay is definitely a challenge even in doing the research, right? We have, once you've run the experiment in your lab, we've sort of suffered like this sort of cross-contamination between runs and between samples. And so that's definitely a challenge. And we've, as a result, had to implement fairly careful sort of protocols, you know, to avoid that. We do have false positives. Just trying to see if I have the data here because I don't remember. I think in our Zika trial, which was 268 patients, I think we had 4 false negatives and 0 to 1 false positives. So we do get them, certainly, but we are going, you know, doing our best to avoid them. And I would say it's one of our top challenges in terms of like just and that challenge, you know, those are data that we get when we're doing tests, you know, hundreds of samples with a sort of for a professional, you know, researcher. And so when we try to leap over to less, you know, like more decentralized testing, we're gonna have to keep sort of innovating around how to reduce that. And then the other part of the question was around cancer, I think.
Cancer, right. Should I repeat that?
No, it's okay. I actually pulled it up. So it's
okay.
So we have not yet looked at cancer. But it's something that I've been, you know, I think decentralizing cancer diagnostics, especially to low and middle income countries where, you know, high costs or high sensitivity techniques are maybe more limited in access, I think would be really interesting. We've just had a streak of outbreaks in the last 6 years with Ebola, Zika, and now SARS that have kept us busy. So, but I think it would be interesting. And a new sensor technology from our collaborator, Alex Green, can do single base pair nucleic acid detection. And that's something that would be really interesting to look at. So thanks for your question.
Great, okay. We have a question here for Galia. Thank you for your talk. Can you resolve, I'm not sure what that, maybe resolve lipid components and things like lipid rafts? I'm interested in PIP2 association with specific receptors. I'm not sure that I understood the question, but if you did,
we're working on proteases, and we don't really look into anything that senses lipids. So PIP2 would not be one of the things that we work on. We usually, I mean, it's an interesting point of view, but we really only work with proteases, not with PIP2 or any of those families. But thanks.
Okay, great. I have here another question. For small peptide drugs, would you still resort to DK4 as a permeabilizing tag? So not necessarily. I mean, it depends on, there's so many cell-penetrating peptides out there. We're actually now looking on this CLIP6 that I didn't really have too much time to talk about, but because it does not penetrate, it seems that it doesn't penetrate cells by endocytosis, but you know, there's so many options. So DK4 is just a simple peptide, so that's why we used it. Okay, I have a quick 2 questions. One is for Keith. Keith, how long does it take to do these glucose-based COVID-19 detections?
So for very like low titer viruses, you know, in the sort of patient like low attomolar range, it takes 2 hours. So there's 2 1-hour steps. If we're looking at things that are more abundant, then even more like 1.5 hours. But this is a challenge, you know, like, so it's competitive with PCR in terms of time. But of course, every time we talk to someone, they're like, they want the answers in 15 minutes or something. So we continue, we have sort of a upcoming sensor technology that we're working on, again, with Alex Green, that can do these low attomolar ranges in 30 to 40 minutes. So we're getting there.
Cool, that's cool. There's another question for, thank you. There's another question for Warren that I kind of missed. Warren, are you thinking of testing for tracking COVID vaccine antibodies responses in Canada?
So the answer is yes. So we've had these discussions with public health and a few other agencies. Some of the issues that we're, again, for home tests that we're still dealing with are things like privacy issues, right? So it's not, the science could be there, but the privacy and other aspect of it. But the bigger picture I just want to mention is that we're actually looking at more of the data, right? It's a way to collect data so that you can understand things that are more than just the assay itself. And so that's where we're headed, right?
Okay, great. I have also a quick question for Katy, and then I'll shut up and let other people ask. So when you showed the PG synthesis, I mean the genetic genes involved in that, Could these also genes be activated in diseases that are not cancer?
Yes, definitely they can. I think that the main, for instance, in obesity, because this inhibitor that I showed, it was specifically developed for obesity.
Okay.
But by using mass spectrometry imaging, we can just find something unexpected and just repurpose drugs, for instance, and stuff like that. But definitely in other diseases, they're also active.
Great, cool. Any other questions from the speakers? Feel free if you have, or any other people who want to ask. I want to see if there's anything.
I have a question for Katy, and it's maybe not the best question, like smartest question, but I'm just trying to wrap my head around how do you calibrate your spatial data to like the real world? So I know you're moving your ion beam, and maybe it's simple, but it just feels like it might be really difficult to get that calibration. And how do you guys approach that?
Yes, so usually we stain the tissue with this histological staining just after we process it, and then we kind of overlay it. And so we know exactly from where the signal is coming from. And we can then tackle a spatial resolution towards.
So you use an image as well.
Interested in separating. Yes.
So there's an image involved.
Optical image.
Okay.
Exactly. Yeah, I failed to mention.
And I guess one last.
Every time we image the metabolite. Sure. Thank you.
And my last sort of question is just like, what's the resolution? Like, how small can you go in terms of resolution?
It depends on the imaging modality. So DESI, which we extensively use, can go down to 50 micron spatial resolution. They claim maybe that nowadays it can go down to 20 micron. I haven't seen it yet, and I'll believe it when I see it. But there is MODI, which can go down to 5 micron spatial resolution. We are getting this instrument soon, and probably then I can tell you if it's working or not. But yeah, it depends on the modality. DESI is much more robust and faster, but it's only 50 microns, which is still good. I started my imaging at Stanford, and there we worked with 200. So it's getting better.
Very cool.
Thank you.
That's
you
great.
So much for asking.
Thanks.
Any other questions to speakers? I just had one last kind of question thrown in the air is, what do you guys think about the field of diagnostics, given this long-term pandemic of COVID-19? How will it affect? If anybody wants to say anything about that.
I think it's very healthy for the field. Like, it's definitely disruptive. It's slightly terrifying that all of a sudden there's now, tripled of people in this space. But I think the field's going to really benefit from lots of new innovative ideas. And I think, I think diagnostics maybe wasn't the splashiest area to be in, until very recently, right? And I think this has maybe made it a little more palatable for investments. Maybe there'll be more startups. I think it'll be interesting. It'll be positive.
Yeah, I think my view is that companies have made a lot of money, and they're going to continue to make a lot of money the next 1 or 2 years in COVID, because a lot of ventures would never invest in diagnostics. You can't make as much money as therapeutics, right? But I think that's starting to change. But all the established companies gonna make a lot of money. All the new technologies you see here, the way I sense it's gonna prep for the next pandemic. The next pandemic could come around next 5 years or 10 years, right? With something else that pops in, right? And so all these things that everyone's developing may not be able to catch some of the COVID sort of crave, because it's gonna take time to translate. But the next one, it becomes a new technologies for the next pandemic, right? It's really about preparation.
I mean, you can say the same thing about vaccines, right? If they made a vaccine in a year, So who knows how fast it's going to be in the next pandemic. It's amazing. It's just amazing. I see that there are a couple more questions. One is for Katy. Could you comment on the sample stability in the mass spec analysis? Would frozen tissue sections work? Is there a time limit? Now you're on mute. Katy, you're in mute.
Sorry. Definitely, yes. We prefer fresh frozen tissues. This is the best tissues for our experiments. And time limit depends. So if it's stored outside of the freezer, then it deteriorates pretty fast, let's say days. But if it's stored in -80, then it can last for years. So definitely, it's pretty robust then. And yeah, and in terms of imaging, it depends on the spatial resolution that you need to achieve. So the higher is the spatial resolution, the longer the imaging takes. And yes, so usually tissue will stay intact through the imaging process. I don't see any problem.
Shayna, there's a question here that maybe you could answer. I'm not sure. It says, are opportunities open for collaboration?
I think it's for me, Eylon. MSIT's mass spectrometry imaging. Yes,
okay.
open.
So I wasn't. Okay.
Definitely open. I'm just starting my lab, but very soon I think we're gonna be running.
Okay. Great.
Forward.
I was trying to figure out what is MSI. Maybe some
mass spectrometry imaging. If I'm not mistaken, maybe they mean something else.
No, I guess you're right. Okay. Okay, cool. Any other comments or questions or no, we're all good. Okay, guys, so thank you so much. This was really, really great. I really enjoyed it. Shayna, do you want to add anything or we should just wrap up things?
Yeah, I'll just add, I agree. It was a great session, incredible talks, each one of them, really different, thought-provoking, great technologies. I think it's also clear that, I mean, you guys are all working in this broad field of diagnostics, but there's so much complementarity in the things that you're doing, but certainly they're also very, very differentiated from one another. So I think it's wonderful that we're going to have the opportunity to work together more and think about maybe putting together some collaborative efforts based on these different technologies. I think there could be some really exciting interfaces between these things that we could explore. So let's keep thinking about that. And I would encourage everybody who tuned in today to also think about that, because there will certainly be opportunities for these collaborations to get off the ground again once we're all a little bit more mobile. So I thank you all for being willing to be part of this event, and thanks to our attendees. And please come back and join us next week for one more session, same day, same time, and then we'll talk more about drug delivery technologies.
Great.
Thank you.
Okay, thanks everybody. Have a great rest of the day or evening.
Thank you. Okay, bye-bye everyone.
Bye.
Bye.